Okay, thanks very much. It's been such a great meeting so far, so thanks to the organizers for the invitation to speak here. So in this session, I'm going to tell you about our cellular flight recorder that we've been using to dissect lineage reprogramming. And really my lab, the core of the lab focuses on reprogramming cell identity. This really is what drives our questions. So we know, from the classical studies from John Gurdon, from Yamanaka, that we can take fully differentiated cells and reprogram them back to a pluripotent state. And from this state, we can then differentiate out to many clinically valuable cell types. In my lab, we're more interested in taking a shortcut on this map. So really, reprogramming back to a pluripotent state and then differentiating is inefficient. It takes a long time. So we like to take a shortcut on this map and transition between two completely differentiated cell identities. So we call this direct lineage reprogramming. It's meant to be more efficient. It's meant to be easier as well. So this transit map of different identities makes this process look very simple, but we know it's not. So we suffer from inefficiency. We also suffer from infidelity. We might be aiming for a particular target cell type. Often we miss, or we make cell types we don't even understand what they really are. So the prototypical cell fate reprogramming protocol that we use is this mouse embryonic fibroblast to induced hepatocyte conversion. So induced hepatocytes were the original target cell identity for this protocol. This was published by Sakura and Suzuki in 2011. So you take your mouse embryonic fibroblasts, overexpress two transcription factors, FOXA1, HNF4 alpha. Within about a week, you start getting these epithelialized cells emerging in culture. And we've adopted this conversion strategy really as our principal protocol in the lab because it suffers from inefficiencies and infidelities. So back in 2014, we published that these cells actually only weakly classify as liver, as hepatocytes, the main cell type of the liver. And there's a mystery cell identity at play here. And we were able to show that these cells actually harbor intestinal identity that was unexpected. The cells also proliferate indefinitely in culture. They can functionally engraft both liver and intestines. So we renamed these cells induced endoderm progenitors, or IEPs, to better reflect this progenitor-like state that we were working with. So this is a really neat reprogramming system, but as I said, very, very few cells successfully reprogram. So this for us was an excellent candidate for understanding at single cell resolution. And this is the team in the lab that have been working on this project, the CellTag team. They've been doing a fantastic job on this project, and they're watching on the live stream right now, so hi guys. Okay, so we take mouse embryonic fibroblasts, we reprogram over a time course of 4 weeks, and we collect cells every 3 to 7 days and analyze them via 10x genomics or DropSeq platforms. So far, over 4 biological replicates, we've collected over 100,000 single-cell transcriptomes via both platforms. So what does the data look like? So this is predominantly, from 2 10x-based time courses here, so over 85,000 cells. We identify 13 clusters of transcriptionally distinct cells. You can see the mouse embryonic fibroblasts down here at time point 0, and over time, our reprogrammed cells emerged. So we've used a method that's previously been published by Barbara, so thank you for sharing this approach with us. There you are. It's been really helpful for us to score cells as a linear combination of either the starting cell type, which are MEFs, and the target cell type, which are these induced endoderm progenitors, so IEPs. And you can see over the time course of 28 days that this induced endoderm progenitor gradually emerges. And when we project these identity scores onto the t-SNE plot here, we can start to identify which cluster is harboring our fully reprogrammed cells, which is helpful. We've identified new markers of IEP emergence in this as well, so ApoA1, which is really useful. And we've been able to broadly stratify the reprogramming process into several phases. So fibroblasts, early transition, and the reprogrammed state. But when we first collected these data sets, we were a little bit dissatisfied because we found many clusters within these transition states. And we were wondering, Are these cells that are trapped in between reprogrammed states? Are they dead-end states? Will these cells eventually reprogram? Because we know it's such an inefficient protocol. The big question was, how do we define these reprogramming trajectories, but how do we identify the origins of reprogrammed cells? So what's so special about the 1% of cells that make it through to becoming reprogrammed? And then how do we use that information to increase the efficiency of the process? So this is really important to us. So as a result of these questions, we decided to develop a clonal tracing technology. So we can trace clones of cells as they reprogram over this 4-week time course. So we call this approach CellTagging. It's based on simple lentiviral transduction. And we need a simple approach because in reprogramming, you've got a very short window of time to manipulate your cells and track them. So if we can throw virus in our cells on top of more virus and track them, it's really easy for us to do this. So CellTagging works with a lentivirus, and just after GFP here in the UTR, we have an 8-nucleotide random barcode. And so we can create a complex lentiviral library of cell tags. We transduce the cells so they express 3 to 4 cell tags per cell. I'll come back to why these combinations are important shortly. We can capture cell identity and lineage information in parallel. So because these cell tags are expressed within the transcriptome at high levels and reliably, that means we don't have to add any extra steps to the library preparation, which is useful. Over 70% of cells pass the indexing threshold so that we can track them over time, so it's efficient. And importantly, reprogramming is not impacted, and also cell physiology isn't perturbed by this lentiviral tagging approach. Now, we go through quite an extensive filtering pipeline. Obviously, we're dealing with random barcodes in a single cell system. You can have errors arising from PCR, also errors from sequencing. So it's really important that we filter out these errors or correct them. We use a whitelisting approach as well to filter known barcodes and remove any noise from the data sets. So when we perform this collapsing and whitelisting approach, we increase the sensitivity and specificity of clone calling in this system. And we're calling clones based on these cell tag overlaps. And this has been a really reliable way of identifying clonally related cells. So what does the cell tag data actually look like? So here I'm showing you 10 clones from 2 independent time courses that were tagged with the same cell tag library preparation. And I'm showing you 10 cells from each clone. So here, 10 cells from this clone, we can see that this clone is defined by around about 10 unique cell tags. So that is the cell tag signature of this clone. Another clone up here, we have 2 cell tags defining it. And you can see some rare examples here where we have 1 cell tag that's shared between 2 unrelated clones. But because we have these signatures, we can reliably call clones independently. So when we look at individual cell tag overlap, we can see that many cell tags are shared between these biological replicates. But then when we look at the combinations of 2 or more cell tags to build this signature, we see that there's no overlap whatsoever between the biological replicas. So it's a really reliable system. Now, why don't we just select or make a longer barcode so that we can track these cells? Why do we use combinations of short barcodes? Well, we see over the 4-week reprogramming time course, in around about 10% of cells of a clone, we do see some cell tag drop out. But the remaining cell tags that make up that signature, their expression is maintained, so we can then reliably track these clones over time. So this really helps the reliability of this approach. Now, the clonal dynamics. So this is using the DropSeq platform, some of our first data sets here. I'm showing you the clone makeup over time. So in this first time point, we've identified 2 clones. And then as the cells divide, we capture them, we replate them, let them grow. We capture again at later time points. We pick up more clones over time. In the second time course here, you can see this massive expansion of a single clone. So when we culture these cells out for a further 5 weeks, the cell line consists mainly of cells derived from a single cell that was reprogrammed, which is slightly worrying, actually. And we don't see this often, but it does happen. Then when we look at the 10x genomics data that we captured, we see way more clones. So first of all, we sequence more cells, but we also capture a greater proportion of cells at each time point. And also, we can also sequence more cells using a 10x platform, so we picked up way more clones here. And now importantly, what we can do is look for the clones that have fully reprogrammed cells at the end of the reprogramming process. And we can start tracking them back to earlier stages and asking, why were these cells different? Why did they make it through and become reprogrammed? In all, we've identified over 12,000 clonal relationships, so it's a really rich data set. Now, a really important question here. So this is not a traditional lineage tracing approach where we can monitor the same cell over time. To measure the cell identity, we're having to remove that cell from the pool, and then we're relying on the remaining cells of the clone representing the identity or having the same identity as the cell that we're measuring. So we test this by looking at correlation between principal component scores. So we use this as a proxy of cell identity or cell state. And we see that clonally related cells have a much more close correlation between PC scores than randomly selected cells here. And we see this across clones. So they tend to have a personality. Some of them are ill-tempered, some of them are recalcitrant to reprogramming, but they tend to have the same reprogramming trajectories, which is a really important point with this technology. So next, how do we use this technology to start building lineages? So this is where our multiplexed CellTagging approach comes into play. So we take our mouse embryonic fibroblasts, we cell tag them with an initial library, and we have a time motif just preceding our random barcode here, so we can demultiplex this information. We then split this pool into two independent biological replicates. This is an important point that I'll come back to later. But then at day 3, we introduce a second CellTag library, and then we introduce a third CellTag library at day 13, so coinciding with phenotypic emergence of IEPs. We can then overlay this data onto the t-SNE plot here based on this motif demultiplexing. And we see our MEF-tag cells here, we see CellTag day 3 coming up here, and CellTag day 3 expression coming up later. These pooled libraries are going to be available on AdGene in about 2 weeks. So AdGene are working really hard on this at the moment. Now, how do we actually reconstruct the lineages? So we use a force-directed graphing approach. So if we zoom in here onto one lineage, we can see in purple our MEF-tagged clones, and then arising from here, we have several subclones that are related by way of this original clone, and these are tagged just after reprogramming, and then in yellow here are the subclones that were then tagged at day 13. So we can build hundreds and thousands of lineages in this way. So it's a really complex data set, and we were quite overwhelmed at first trying to identify successful reprogramming trajectories from here. We have this data available on a website. So this is CellTag Viz. So you can find this at celltag.org. This is inspired from DropViz from Steve McCarroll's lab. So imitation is the sincerest form of flattery, I think. So if you have any feedback from this, any suggestions, if you want to do any analysis for us, then we'd be very happy to hear about that. So breaking down this complexity from thousands of clonal relationships, how did we initially do this? So we reprogrammed the cells so we could gain higher resolution information from these later stages of reprogramming where we had the highest clonal resolution, so from around about day 6. We then identified the fully reprogrammed cluster here, and we use a randomized test to identify clones that were either enriched for fully reprogrammed IEPs, or they were either depleted of fully reprogrammed IEPs. So these were the kind of super clones that are really good at reprogramming, and these are the guys that really just do not want to reprogram whatsoever. We then can overlay this information onto the t-SNE. So we use a contour plot here to see where these clones are actually landing. So we define this as a dead-end trajectory. These are from the IEP depleted clones. We can also link the areas of highest clonal density from day to day to create these trajectories. Then when we look at the opposite, so these super IEP enriched clones, we can define the reprogramming trajectory. So you can see here that it largely really doesn't overlap much with the dead-end trajectory. So it was really nice to see such clear definition between these two different reprogramming paths. We then compare this with pseudotemporal ordering using Monocle 2. So we use semi-supervised ordering here based on ApoA1 expression. we can see two clear branches of reprogramming. And then when we overlay clones from the dead-end trajectories and the successfully reprogrammed trajectories, we can see a nice split between these two different paths. So there's some nice agreement there with Monocle. So then one of the questions that we can ask before we start looking at the molecular flavors of these two paths is reprogramming deterministic or stochastic? So only 1% of cells will reprogram. People, based on that number, have said, well, it must be stochastic, seeing as so few cells reprogram. So what we can do with these lineages is we can identify clones in the same lineage that are derived from a common ancestor. And then we can ask these related clones, so they've been tagged just after the reprogramming process has ended with the transcription factor overexpression. Do they reprogram with different efficiencies, which would support the stochastic model, or do they reprogram with the same efficiencies, which would support a deterministic model? And we were thinking, okay, we've had a lot of evidence for stochastic reprogramming in the literature. There's also evidence supporting deterministic models. But we were really surprised that clones that are derived from the same common ancestor reprogram with the same efficiencies, especially those clones enriched for fully reprogrammed cells. So this was really surprising to us. Now, I said earlier that we tagged that original MEF pool, and then we split and reprogramming two independent biological replicates. So using that experimental design, we can ask, is there any elite or privileged cell type in the highly mixed MEF population that's predisposed to reprogram? We don't find that whatsoever. So we find clones that are related to each other, so sisters from each biological replicate. One sister will reprogram successfully, the other sister nothing, or reprograms with a different efficiency. So this is suggesting that there isn't an elite cell type in the original cell population that's predisposed to reprogram, but there is a privileged state. So cells within a short space of time, if they're in the mood to reprogram or they have the personality to reprogram in that time, then they will reprogram in a deterministic manner. Okay, so just to wrap up in the last couple of slides now, we've been able to define these two trajectories. We can define whether or not this process is stochastic or deterministic based on these trajectories. We can now ask, what are the molecular differences that lead to this bifurcation? So when we look at this identity scoring again, between the reprogramming and dead-end trajectories, we see that over time, we have a gain in IEP identity over reprogramming trajectory, and then we gradually lose this IEP identity and start moving back toward a fibroblast-like state on the dead-end trajectory. And when we look at the kind of significant gene expression differences between these two trajectories, we see that fibroblast gene expression gets reactivated on the dead-end trajectory. We also see activation of imprinted gene expression, which I think is really interesting. On the reprogramming trajectory, we were really interested in this expression of a putative methyltransferase, so metyl 7A1. There's really not been a lot appearing in the literature about this gene. It's related to metyl 3 and 14, so methyltransferases that are associated with ES cell differentiation and reprogramming to pluripotency. So naturally, we're wondering what this gene is doing. And we see it's expressed around about day 21 on this reprogramming trajectory. So it's transiently upregulated. We don't see this upregulation on the dead-end trajectory, where rather the cells go into this fibroblast-like state again. So the final part of the story here is that we take our standard reprogramming cocktail of FOXA1, HNF4 alpha, we add metyl 7A1 to this cocktail, and we see a consistent increase in the number of fully reprogrammed colonies formed in this assay. Then when we look at single cell resolution with the standard cocktail and then the metyl 7A1 enhanced cocktail, we see way more cells in the fully reprogrammed state, so suggesting this is enhancing the reprogramming process. And we have lots more candidates to test in this way as well. I think if we can get back to those origins, find out what's happening in these cells that are programming in a deterministic way, then that's going to throw up some interesting candidates. So in conclusion, I've shown you our cellular flight recorder today, our cell tagging system. It's enabled us to identify these successful reprogramming trajectories. In this IEP generation, the successfully reprogrammed cells are derived from a handful of initial cells, and then they expand through the reprogramming process. We see the fully reprogrammed and dead-end states. We've been able to characterize these states in molecular terms and also enhance the reprogramming outcome based on this. And finally, reprogramming outcome seems to be determined from the outset of transcription factor expression by the state of the cell that receives the transcription factors. And I think that's going to be really exciting to follow up on. So finally, I'd like to thank the lab, who are an amazing group of people, our funding bodies, Barbara for the quadratic programming help, thanks very much. And we are currently hiring postdocs. I've been censored here. So yeah, we're looking for postdocs to join this fantastic team. So if you're interested, please reach out to me. If you know anybody who's looking to do a postdoc, then put them in touch with me. So I'll be happy to take any questions. Thank you.
Questions?
Hi, Sam. Great talk.
Thank you.
I was wondering, when you have these IEPs and you engraft them or co-culture them in a liver environment, say, do they become more liver-like? Does the whole population follow in a
yes.
uniform
trajectory?
So
the cells are in a progenitor state, and depending on whether or not you transplant them into liver or intestine, transplant them into liver, they will respond to that niche and they will differentiate into liver. Transplant them into intestine, they will differentiate into intestine. And they seem to form all the different cell types of the intestine as well. And we think that's from an LGR5 stem cell state. So can they functionally engraft other endoderm organs? We don't know. We haven't tried yet, but we'd like to try.
Thank you.
That was very nice. You showed the MET-L7A comes up rather late in the trajectory. Do you have an idea? Are there deterministic factors earlier on as well that are separating these?
Exactly. That's a really difficult question. So it's easier to look at these later bifurcation stages. So we can increase the efficiency around about 2 to 3-fold by targeting genes that are upregulated in these later stages. I think if we could somehow find the genes that are driving these differences in these early phases, we could potentially create a deterministic reprogramming strategy.
Can we deplete the reprogrammable cells by repeatedly challenging them to differentiate, or after one round they're either really dead or
we've been thinking about that. So can we enrich that dead end population and push them over into a successfully reprogrammed state? But finding the factors that are expressed in these early stages is difficult. So we think these reprogramming events are coming from single cells, so they're individual events. So if we capture those events and we can measure them, those cells will never reprogram. So I think it will take an imaging approach to really crack this.
Yeah, so I missed how you so you say that the successfully programmed cells, they have a shared history, basically. So what is the control for this statement? So compared to what? This is more than.
Can you repeat your question?
Okay, so you say that cells which are successfully reprogrammed, they tend to come from the same origin cell, correct?
Yes.
So now let's say you look at the dead end cells.
Oh, okay, so the way you're looking at this.
That is a difference, or to random samples, or what are you comparing to?
We compare this to random sampling.
But that's maybe not the right...
So in this context, though, we are looking at cells, so we define the dead-end trajectories and the reprogrammed trajectories, and then we look at the percentage of each clone that's either on the reprogrammed trajectory or the dead-end trajectory. And we see that those percentages are the same for all the subclones. So we haven't done any permutation testing to see if this is by chance, but it's every single subclone that's following the same trajectory. And I think an important control there as well is taking that initial meth population and looking at the clones across the different biological replicates. And there we actually have the physical control where we see that they do not reprogram with the same efficiencies when they've been physically separated. So I think that gives us an important control as well.
So the statement is that this linkage is specific for the reprogrammed cells.
That we haven't tested.
Yeah, right. But that's I think that's really I think my point.
Yeah, I see what you're saying. Yeah.
Okay, one last question.
Hi, great talk. My background is RNA biology, so I got very excited about M6A. And one thing is that M6A turns out to also be the pathway by which the Zika virus very specifically affects neural progenitor cells in the brain. Mm-hmm. And so it's interesting to me that there's this overlap here in programming, reprogramming, and M6A is notoriously difficult to measure because you have to use an antibody, you have to use a lot of starting material, so it's super hard at the single cell level. And I'm wondering if you're planning to maybe instead use some sequencing technologies, PacBio or Nanopore, to try and see if you can measure M6A. I know it's going to be still really difficult, but it will just be a few transcripts that'll be interesting, but I think those are going to be the key ones.
Exactly, and that's something we would really love to go after in terms of this mechanism. So now we have the phenotype, which is always great to start with, but next will be the mechanism.
Great, thank you.
Thanks.
Right, let's thank Samantha one more time.
Thanks.